All right, everyone. Hello and welcome to GLOBEBUSTERS, Season 15, Episode 9. Today, Tayler is going to give a presentation on Abiogenesis, a complete review. But before we get into that, let me introduce everyone. I'm joined, of course, today by Stone G, Shane G, and Tayler G. Welcome.
Yo, hey, yo,
And I
hey.
think her husband Dan may be chiming in as well on the presentation. It might be a little duo effort here. All right,
Absolutely.
So yeah, welcome Tayler. Welcome Dan.
Welcome, guys. So I've presented this before on Ether Cause and Earth Awakenings, but was asked to come present on GLOBEBUSTERS. I think it is a very important topic. It shows just how things that the scientific community will portray to the public as facts. Once you actually look into the science of it, it gets a little bit messier than that. So I'm going to be breaking down the origin of life. So I study biology, as does my husband. And whenever we started studying biology and looking into it, we found that, life does not seem to appear to just come about by accident or undirected design or anything that's undirected. So chemicals will actually move away from life instead of to life. And I'm going to be getting into the background of the biology and the chemistry as to why that is so. All right, so my presentation is titled, Does Science Suggest Abiogenesis or Intelligent Design? All right, I'm going to give a little bit of background into biology. So biology is the study of life and living organisms. Cell biology is the study of cells, and this is what I study. And cells are the basic fundamental unit of life. And the word cell is actually derived from the Latin term cellula, meaning small room. And there's a lot of different branches of biology, such as parasitology, biotechnology, histology, physiology. And they all kind of branch back to cell biology because cells are the basic unit of life. And again, this is what I study in particular. All right, so here's some fundamentals of biology. Characteristics of life are that they are made of cells, made of one or more cells, and this is why what they call viruses are non-living. Life displays organization. Life is able to grow and develop, reproduce, respond to stimuli. It requires energy and maintains homeostasis. So just a little hierarchy. So you go from molecules to organelles, which is within the cell, to cells. Cells make up tissues. Tissues make up organs and organ systems, which make up organisms, populations, ecosystems, and the biosphere, which encompasses the ecosystems on Earth. All right, so single-celled organisms, people have an idea, and back in the day, they used to have an idea that they were just like these balls of jelly. But now that we can actually look with microscopes, we see that cells are much, much more complex than you might imagine. So here's a picture that I took of Basilaria paxiferia, captured by me. So these are some cute little diatoms called the dancing diatoms, which kind of dance around one another. And then on the right here is a little protist, and it looks like it almost has a little heartbeat. So these are just some intercellular interactions that are occurring within the organelles. And then in the center here, believe it or not, these are actually real organisms, real diatoms that you can find in a pond. And this gentleman, Claus Kemp, he actually arranged them and makes art out of them. So yeah, diatoms can look very amazing. So they have a silica shell, which makes them look really pretty. And so this is a single-celled algae, and it's called a diatom, which is a type of protist. Just some little fun facts here. All right, so this is microscopic images and videos of human cells. So here are neurons here on the left trying to make connections. And then in the center, this is a human U2O cell dividing, and you can see there is M. Cherry labeling the tubulin, which is pulling apart the chromatin. So you can see cellular division actually happening here. And so this is using fluorescent microscopy, where basically you attach a fluorescent protein to the protein that you're trying to track within the cell. And here they labeled chromatin and tubulin. So you can see how the cell is pulling the DNA apart. All right, what I study is microtubules. So these are some videos on the right captured by me of growing microtubules in Chlamydomonas reinhardti. So we basically have a fluorescent EB1, which is at the growing tip of the microtubules. So we can actually track the microtubule growth, or I track the microtubule growth within Chlamydomonas, just to see how microtubules in the cytoskeleton are affected by external toxins like aluminum. So I'm studying how aluminum is harmful to the cytoskeleton, which is kind of a controversial topic in biology because, you know, aluminum is used as adjuvants in vaccines. So a lot of people say that aluminum isn't harmful, but I'm trying to show with my research that it actually can alter the cytoskeletal dynamics in the microtubule growth. All right, so fun fact, microtubules are attributed to fiber optic cables and neurons, and biophotons are a new theory of how neuronal signals are transmitted, not just through chemicals, but by light. So just a little fun fact. All right, so here's the intercellular components, and understanding the complexity within the cell is very important. Excuse me, I have like a sore throat. It's very important to understand how difficult of a task it is for a cell to arrange on its own. All right, so in the upper right-hand image, this was created by Dr. McGill, the director of molecular visualization at Harvard Medical. And he said, quote, although people are struck by the complexity and density of this environment, what we depict is actually very dilute in its concentration relative to the cell. I'm so sorry, guys. Excuse me. So what we're seeing here is like just the intercellular complexity within a cell. And then here's some electromicrograph images of different organelles like the Golgi, chloroplast, ER, endoplasmic reticulum, and the mitochondria. All right, so cellular composition. The reason why I'm telling you this is because I'm going to go over how these things could have formed. All right, so we have amino acids, which make up proteins, which are all left-handed. We have fatty acids, which make up lipids, which are mostly right-handed. And I'm going to go over chirality in a second here. We also have sugar molecules, which make up carbohydrates, and nucleobases, which make up DNA and RNA. So all of these have what we call chirality, meaning they cannot be superimposed on each other.
Hey, Tay.
Mhm.
Maybe you could break down what you mean by left-handed and right-handed for the audience and us.
All right, so oops. So what I mean by that is if you see the molecule right here, you can see like kind of like our left and our right hands. When you line them up, you have a thumb that sticks out on either side. It's not like we have two left thumbs or two right thumbs. And molecules are the same exact way. So in nature, whenever you form things like amino acids, they will naturally form in their left and right-handed form, just like we have both of our hands. But however, in life, they must all be left-handed. That means we all need to have hands with left thumbs for them to interconnect. And the reason why we need homochirality is so that we maintain structural integrity and preserve function. So amino acids make up proteins. If we had left and right-handed amino acids, the proteins would break down. And in nature, in undirected design, you have amino acids that both form left and right-handed amino acids. Whereas in life, we only need left-handed amino acids. So this is one of the big hurdles to overcome when thinking about how life could form on its own. Does that make sense?
Somewhat. You touched on the geometry of it just at the beginning, and then you moved on from that definition. So we're kind of getting it.
Yeah, so chirality means that they can't be superimposed on each other. Just like our hands. Does that make sense?
Are we saying
Yeah,
that
so
nature
the way
produces?
that the go ahead.
Sorry, I was like saying that nature produces left and right, but we only need left. Is that what you're saying?
Yes.
Okay, that's
And
weird.
scientists are trying to figure out how exactly this could be the case. Like, how could have nature produced only left-handed amino acids and then right-handed nucleotides? You know, this because in nature, whenever we just take the chemicals and put them together to form amino acids and nucleotides, which nucleotides make up DNA, they form both left and right-handed, just naturally. But in life, they would have to all be either left-handed or right-handed. So there's a huge hurdle to overcome.
Huh.
Nice. In your slide there, it has like the H represents what the thumb would be. What is the H in that structure?
Hydrogen.
Hydrogen.
Mhm.
So the binding part of this molecular structure like has like a preferred facing pole, you could kind of say. So
Yeah,
left or right.
exactly.
Yeah. Okay.
Yes, that makes sense.
Gotcha.
Yes,
Gotcha.
they're trying, and they've said they're trying to propose ideas like, oh, well, maybe it could have formed on crystals to make them all left-handed or right-handed. So it's funny to watch them kind of squirm around, try to figure out how this could have been so, because in nature, what we see occur just wouldn't be compatible with life. All right, so I think this is one of my last background slides. And then so just to understand the complexity of the cells, so people say, oh, well, in the early Earth, they could have formed simpler cells, right? But let's look at the simplest cells that we have here on life. So the smallest cell is Mycoplasma genitalium, and it's in the reproductive tract of humans, and it is made of 100 billion atoms. And one yeast cell has 42 million protein molecules. The shortest DNA in the Mycoplasma genitalium has 580,076 base pairs, okay, which would be 34 million atoms. And the shortest DNA strand is 507 genes. Now, they took this simple celled organism, the simplest, what you'd call like the simplest life form on Earth, and they actually tried to remove as many genes as they could to see how many it would need to function. And so they took it from 507 genes down to 438. So, and they found that once you started removing more, the cell just basically couldn't function. So the minimum number of proteins found in the simplest known life form is 438 genes. And genes are large segments of DNA of nucleotides, which code for proteins. Okay, so the genome of this bacteria has 531,490 base pairs and 438 protein coding genes. So that's just to give you an example of what the simplest life form would require. And then just the basic components of life are oxygen, carbon, hydrogen, and nitrogen. We also have phosphorus, sulfur, potassium, magnesium, and zinc. So these are all required for life. And there's a funny thing with phosphorus. I have a slide on it, and NASA actually. So we'll get to that in a little bit. NASA basically got called out for lying about a certain research project on phosphorus. So anyway. All right, so cell functionality. How does it all work together? Where does it store information and how does it use information? We're going to look into this. So the cellular information system undergoes a central dogma of biology. So the central dogma is basically how the cellular information system works. So it goes from DNA, which has like the instruction for life, to mRNA, then to proteins. And this is a very complex process. All right, and on the right here is a picture of DNA, of E. coli DNA. And this is in a scale, you can see a scale right here of 10 nanometers. All right, and I have citations down here if any of y'all want to check this out. But a fun fact about the central dogma. So the term dogma was a misnomer as it implies an unchallenged belief, which contradicts the scientific method. And Crick later, himself later acknowledged this improper use of the word, but we still use the word central dogma, even though dogma is a misnomer, because in science, everything should be challengeable. You should be able to test and challenge any idea in science. So you don't want to be dogmatic in science, as things are up to revision and need to be repeatable and testable, so on and so forth.
And nobody figured that maybe the word like postulate might work instead. They just had to stick with dogma.
Well,
Yeah, I feel like Crick knew exactly what he was doing.
yeah. Yeah, exactly. So we just kind of stuck with the term, even though, because that's what it was originally called. But yeah, things should not be called dogmatic, as they should be able to be challenged and tested. For sure. All right. So original life, was it a random cosmic coincidence, as Gally says? I love that term. So was it just something that happened undirected? So the scientific community hypothesizes that life originated through a random undirected process known as abiogenesis. Abiogenesis is the idea that life arose from non-life. And by the way, we have never been able to make life from non-life in the lab, even with the best possible conditions. Even if you take a cell and let's say you take like an amoeba and you poke it and the intercellular materials flow out of it, you can leave it there forever and change the pH, temperature, whatever you want. That has never been demonstrated to be able to form an original cell, a living cell back again. Even though all the materials are there and put together for it, like we cannot, make life from non-life, even from just putting the organelles together. So it seems like it's a difficult task to like get it to start going and sparking. Even if you put everything together, that doesn't make it all the processes start functioning. So even just having the basic material there, it doesn't even really solve the problem, because you can have everything there, but getting it to start going is another thing, because the cell has a lot of intercellular components that are working together all at once, and it's actually amazing. I think Harvard has a video that tries to demonstrate life in the cell, and you can just see all the working parts. It's like a fully functioning like city working together, all doing their own proper task. All right, and then a fun fact, panspermia is the concept that life may have originated elsewhere in the universe and then reached Earth through like space travel and arrived here on a comet. But that's like just pushing back the problem. If there is carbon-based life, this would apply anywhere in the universe, in the mainstream model of the universe, as to how life could form from non-life. So this simply just pushes back the problem to say, oh, well, it could have came from some other planet. And Watson and Crick actually proposed this idea after discovering the double helix shape of DNA. And they said, wow, they said, this looks incredible. It couldn't have formed on its own. So they thought aliens may have created it or something, and came up with the idea of panspermia, or this could have been some extraterrestrial life form. So yeah, I think they were atheists. And so they tried to say, they knew it was amazing, but they just said, oh, well, this could have just came from somewhere else in the universe. But
They
anyway,
knew it was
panspermia.
amazing, but they didn't want to be dogmatic about it, right?
They definitely didn't want to give an intelligent design any credits, intelligent designer, because I believe that I've heard that they were atheists. So yeah, they just proposed panspermia instead of intelligent design. All right.
Classic.
So here's the history of abiogenesis. So in 1859, Darwin proposed the theory of evolution in his book On the Origin of Species. And in 1870, the term abiogenesis was coined by Thomas Huxley to describe the process by which life could arise from non-living matter. In 1871, Darwin described the possibility of life emerging in a warm pond. So, from what I read and understood, they were basically, once Darwin came up with the origin of species, trying to explain how speciation occurred through evolution, they were pressuring him to try to come up with how life could have formed. So basically, just like the universe expanding, reversing back to hypothesize that the Big Bang started the universe. Basically, you take life forms and you reverse it back. Oh well, life must have popped into existence somehow to start off the evolutionary process. So they're kind of pressuring him to come up with a theory on the origin of life, and this is how like the warm little pond hypothesis came about, that life could form in a warm pond. And this was in 1871 by Darwin. And then in 1929, Operin and Halide proposed that life originated from a primordial soup, which is a mixture of organic compounds in early oceans energized by lightning or UV light. Now they've since like refined this, because in the ocean, basically, you would have just had hydrolysis occur. So now for the original life, they need wet and dry cycles. So you can't have like proteins form and other molecules form in water, because they would just hydrolyze and break apart. And like for protein formation, you have like the removal of a water molecule to make the amino acids bind to form protein. So if it was underwater, that wouldn't be able to occur. So now they cope with this with wet and dry cycles, so it couldn't have just happened like in the ocean.
So it
But
would
that's
have
a
to
side.
there would have to be a small pond near a desert somewhere or something, so that they could concurrently evolve.
So they actually say in the walls of a hydrothermal vent where they have basically micropores in rocks in a wall of a hydrothermal vent. So now that we understand what conditions you need for proteins to form and what conditions you need for, nucleotides to form, you have to have like pretty specific conditions. So for proteins, you have to have a very basic environment, and that would only happen in a hydrothermal vent. And then also you need wet and dry cycles, so it'd have to be in the pore of a rock of a hydrothermal vent with a very low pH.
Wow.
And it gets really messy. It gets even messier than that, but
With a fox
that's
in a
just
box
for-
on a train in the rain.
Yes, exactly. That's exactly what it is. And that is the current state of the field as we speak. All right. So let's see. All right. So they have right here abiotic synthesis reactions on the early Earth. This formed molecules for the prebiotic soup, and also organics from space, which they literally still invoke in modern day papers, had to come in and also contribute to his prebiotic soup and form prebiotic polymers, origin of life, RNA world, and then the protein DNA world, modern biochemistry. So they basically say, yes, from soup came polymers, polymers formed life, life formed RNA, and then RNA could have formed proteins and DNA. But this is very speculative at best, as we have not replicated this whatsoever. All right, so let's talk about the knowledge of the cell at the time that they came up with this theory. So whenever they came up with the idea that life could arise from chemicals, I was trying to find some writings on the cell around the same time. And I was able to find this excerpt from Fortnightly Review, which is a popular magazine in 19th century in England. This was written by George Lewis in 1868, titled Mr. Darwin's Hypothesis. All right, so here's a quote. The simplest form of organic life is not as commonly stated a cell, but the microscopic lump of jelly-like substance or protoplasm. The definition of a cell is a nucleus with surrounding protoplasm. The cell wall or delicate investing membrane that which makes the cell a closed sac is no longer to be regarded as a necessary constituent, but only as an accessory. So this kind of gives you an idea of what they thought about the cell at the time, a nucleus with surrounding protoplasm. So as I mentioned earlier, they just thought it was like a lump of jelly with called protoplasm and nucleus. And that's basically the extent of it. They weren't able to dye things and see things under high-powered microscopes to actually see all that's going on in a cell. So it might be easy to conceive that life can form just from a chemical soup if this is all you think that life is. So, yes. They didn't have any real understanding of the cell at the time. So, 70 years after abiogenesis, was somebody going to say something? I'm sorry.
Well, to me, it seems like they were suggesting something parallel to how maybe an endoskeleton doesn't really need a dermal layer to hold the organs in.
Right.
That kind of hilarity.
Yeah, it was just super simple knowledge and understanding of the cell at that time. All right, let's see. So abiogenesis continued. So it was 70 years after abiogenesis was proposed that we could actually begin to see the intricacies of the cell. So in 1940, Manfred von André Ardenne constructed an electron microscope, which greatly improves our ability to visualize cellular structures. And in 1945, the first electron micrograph of an intact cell revealed the subcellular structures. And that was in 1945. So this was published. And so now you can see things like this is the flagellum. And then we have the mitochondria and other organelles that you can see better. Also, fluorescent microscopy, which is what I work with to see the cytoskeletal dynamics, that was also created much later. So you can actually track the intercellular dynamics happening, kind of like I viewed the microtubules growing in the cladomonas. So we had advancements much later, which can actually allow us to like see what's inside the cell. Also, you can just introduce like dyes to living cells and view them under high-powered microscopes. And the dyes will stick to different components within the cell. And so you can actually see these structures much better, even without using an electron microscope. And
Just to let you know, Tay, just to let
yeah.
you know, as you're going through your slides, that we can't see your mouse. So when you say this, you have to describe where you're pointing.
Oh, really? Thank you for telling me that. So yeah, the flagellum is in the upper left picture. Mitochondria is in the upper right. And I'm not 100% sure what this one is, but it's just different organelles within the cells. And they between different types of cells, they might look different. So yeah, you can basically just see that the cell is extremely complex and dynamic with higher powered microscopes. Okay, so I was looking into the current state of knowledge on abiogenesis. So I was trying to see like what universities use the tech as a textbook and what these textbooks say on the current state of knowledge in abiogenesis. And actually, it's very brief. In the Campbell's biology book, I was only able to find like 1 or 2 pages on the origin of life. So it's like they're trying to gloss over it very briefly, probably because they know that it's not reproducible. And even within abiogenesis researchers themselves, they can't even come up with a model or an understanding of how the basic molecules formed and what conditions they formed, in what order they formed. They're still deliberating on these very basic fundamental components to the original life. So we're just trying to figure out which molecules came first. Was it proteins? Was it RNA? I've debated people who thought it was both, either. Like, so I've debated people who thought it was protein first through like amyloid synthesis, and then I've also debated people who believe it was RNA first, and also people who think it was metabolism first. So they're still in heavy disagreements within themselves on how the original life came to be. So that's probably why they're not in textbooks, and there's not much in basic biology textbooks, just because they can't really come to a consensus, and a lot of it isn't reproducible. They can reproduce small, tiny steps of it, but they can't reproduce a coherent like model to show how these biopolymers would form. All right, so this is all it basically mentioned was the operin halide experiment. We mentioned that one and the Miller-Urey experiment. So I'm going to go over the Miller-Urey experiment, but this to this day is the most cited evidence for life being able to form from chemicals. All right, so here is the Miller-Urey experiment that is mentioned in modern day biology textbooks as evidence for the origin of life. All right, so this experiment occurred in 1953. Most cited evidence for abiogenesis. So what they produced was a 2-page long paper, which posited to give credence to the primordial soup hypothesis. So what they did was they attempted to simulate early Earth's conditions by mixing gases such as methane, ammonia, and hydrogen in a sealed chamber. Then they added water and heat to create vapor, simulating oceans. Then they applied electrical sparks. So they had water and then they basically vaporized it, meaning that evaporation maybe could have occurred. And then they applied electrical sparks, such as lightning striking this vapor. And then they cooled it and condensed it to simulate rain. And then they collected and analyzed the tar that was produced at the bottom of the flask. And you can see the image here on the right, bottom right. So yeah, so basically you have water and then evaporation occurring, getting struck with lightning, then condensation happening, it raining, and then we collect the water droplets at the bottom, and then we basically, as a result, get this tar-like substance. So this tar-like substance was analyzed, and this is what they found. So they used paper chromatography, which is used to separate different types of molecules, and they basically found aspartic acid, glycine, alanine, and butyric acid. So glycine, beta-alanine, alanine, and these were the 3 major amino acids that were formed. And I should mention that they were also formed in left and right-handed configurations. But earlier, remember I mentioned they all have to be left-handed. So even this would not have been usable for life. But this is not that impressive because like they put in, let's see, they put in they put in H2O, hydrogen, methane, and CH4. So they basically put in chemicals, sparked them, and then came up with these substances, glycine, beta-alanine, alanine, which are amino acids. And so here's the reaction right here. Methane, ammonia, and hydrogen. And what they produced was around 10, 13, and 13 atoms. So it's not very impressive. Like, to me, this is not very impressive. And it's only 2% of the actual tar was the amino acids. So 2% of the tar was amino acids. And these amino acids are both the left and right-handed, making them not usable for life, unless there was some mechanism that could occur to make them all left-handed, which they're still trying to figure out how that could have happened. So not that impressive to me, and unusable for life. And so in comparison, there's about 100 billion atoms in the smallest cell, and what they produced was things that were around 10 to 13 atoms. All right, and they were left and right-handed, not usable. All right, so people have posted criticisms of the Miller-Urey experiment, such as Dr. Scott Hughes. So a couple of his criticisms were that concentrations of methane and ammonia were carefully selected. So there was like intelligent design put into this experiment, but that's a side point. So there's no evidence to indicate the early Earth's atmosphere was reducing. This is debated, but you know, it would have to be for life to form. So of course they're going to say it was reducing. And also, the reducing atmosphere would have been fatal to life forms. So lightning would have destroyed any organics. Molecules produced would react detrimentally to life forms. So there was a lot of chemicals in the tar. They're only focusing on the 2% of amino acids, but there was a lot of other harmful chemicals to the tar.
I can see them leaning with the lightning. They could weasel their way onto that one. You know how the mainstream love to go, well, it's sort of affected by it, but they don't actually quantify it. This one they could say, well, maybe those cells were near where the lightning went down and didn't get vaporized like the rest of the stuff. And it sort of got a bit of lightning. And can't you just hear that coming?
Yeah. So basically, to form the amino acids that they formed, it would have had to have been struck by lightning. But think about how many amino acids and trials that you would need to get all left-handed and to actually get a functioning protein.
Yeah, but
There's like
Frankenstein got
on
hit
the
by lightning, right? That makes total scientific sense. That's
Hell
probably
yeah,
why
but
it's
he had
in
a bolt in
there.
his neck first.
Oh yeah, right.
Yeah, so like with the whole lightning thing, there would have had to been a lot of lightning occur and then form a lot of amino acids to have a lot of trials to where you only have left-handed amino acids form a protein in the correct environmental conditions with the instructions that would somehow be needed to form more proteins. It's such a crazy mess, to be honest, but they also mentioned how formaldehyde and cyanide were formed by this reaction, which are destructive to life. But we're just going to focus on the 2% of the few amino acids we formed in the tar. Like, forget all the other harmful stuff that was produced. We'll just separate that out and throw in the trash. So anyway, did Miller-Urey imitate early Earth conditions? So here was some quotes from some papers saying that it's possible that primitive atmosphere was not as reducing as the Operin, Urey, and Miller believe. So the accepted picture of early Earth's atmosphere has changed. It was probably oxygen-rich with some nitrogen. I wouldn't say oxygen-rich. It could be more oxygen-rich than they originally thought, but this is still debated. And they also say it could have been a less reactive mixture than Miller's, or it might have been composed largely of carbon dioxide, which would greatly deter the development of organic compounds. So models suggest that there were significantly low concentrations of NH3 in the oceans, and similar experiments in oxidizing environments yielded no results. So they were actually like the concentrations of the organics, like they were like obviously using higher concentrations of methane, ammonia, and hydrogen to optimize like the results, to try to get the results they wanted. But, you know, how do we really know what the early Earth's environment could have been? I mean, they can try to look down in rocks and see, was there a lot of methane and stuff at the time, but they're just kind of guessing, and they're also intricately putting in what they want to get the specified result.
Oh, they're
And
even
also,
guessing about the age of the rocks.
yeah, maybe. But my main contention with it is that, you know, even the amino acids that were formed were left and right-handed, they weren't usable. And then there's also the tar problem, where 98% of it wasn't this, and it probably would have locked up any of these amino acids that were formed from even being used for life. And the tar problem was mentioned by other scientists later as well. Okay, let's see. So did Miller-Urey imitate early Earth conditions? In Urey's book, The Planets, Their Origin and Development, it stated the primitive oceans would have been approximately 10% solution of organic compounds. This would provide a very favorable situation for the origin of life. So they were very wrong. So the current organic matter content of oceans, of ocean sediment, is approximately 1 to 3%. And this is with life thriving in the oceans. They were hypothesizing 10% organic compounds in the ocean. So they were like
Yes.
giving it very favorable conditions. Can you believe? So yeah, they were saying 10% organic compounds would have just been floating around in primitive oceans. Even in modern day oceans, we only have 1 to 3% in sediment. And this is with current research that I was able to find. So yeah, they were tweaking it just a little bit and putting in whatever that would be needed to get the amino acids, of course. All right, so here's a genetics researcher, Albert Bransom. Here's a quote from him. We claim that living systems evidence a preoccupation with maintaining chemical specificity, having not just the right molecules to hand, and especially not having all the wrong ones. Yet in any solution chemistry or any chemistry not managed by a macromolecular machine acting specifically to affect the particular transformation needed, the latter wrong ones invariably overwhelm the former right ones. Okay, so this is what happened with the Miller-Urey, because we get only 2% of the reaction actually formed these amino acids, only 2%. But think about the other 98%. In most of these chemical reactions, you get a lot of stuff that we don't want and could actually be harmful to life, and only 2% of stuff that we do want. So that was just a quote by him. And here's the tar problem. So origin of life researcher John Sutherland, so he actually researches origin of life and tries to find ways that life could have formed from chemicals. This is a quote by him. The building blocks assumption is simply dead wrong. A simple thought experiment should suggest if you make up any mix of building block products you like, however rich, energize it in any way you like. Wait as long as you like. Nothing we assert having any claim of any sort to being lifelike will ever arise in your flask. Not ever. Instead, the chemistry therein will be more or less slowly progress towards Benner's tar. So, Origin of Life researchers, John Sutherland is basically saying, if you just put in these building blocks, you can energize them however you like, wait as long as you want. It's going to move away from life and not going to give you anything lifelike. What will come out of your flask? Nothing lifelike will come out of your flask. Instead, chemistry will more or less slowly progress towards Benner's tar and just form a tar-like substance, which actually would hinder life's formation. So this is the building blocks assumption that he's calling out, saying that it's dead wrong. If you have these building blocks, they're just going to form like tar. They're not going to form anything usable for life. All right, so now my next quest was to search for a concise model for abiogenesis. So if you remember earlier, I was mentioning all the different models for abiogenesis. We got the RNA first, we got the proteins first, and so it is still messy to this day. So here was a current paper I was able to find, and here's just some quotes from it. So they invoke extraterrestrial sources for organic materials such as asteroids or comets. It says that ongoing research on plausible prebiotic synthesis could be in cyanosulfidic environments and alkali hydrothermal vents. And also the amino acid abundances from our asteroid samples return will soon contribute to our understanding of it. So they're still trying to understand how asteroids could have maybe brought in the organics, and they're imposing all these different crazy environments. And they also mentioned, as I mentioned earlier, the water rock interface of Euclides's subsurface ocean, which is to be alkaline, which is to be next to terrestrial alkaline hydrothermal vents. So they're invoking the pore of a rock in a hydrothermal vent. And this is just for protein formation. This is not for RNA formation, because RNA cannot have formed in these conditions. And then they also mention how perhaps the biggest mystery is how sequences such as the common ancestor LIV-tRNA synthetase would have arisen, which would have translated the genetic codes to become from DNA to RNA to protein. So how would this machinery that could translate things from DNA to proteins, how could that have arisen? It's literally like a translating system, which I have a few slides on later. But this ancient tRNA synthetase is a huge question. And it's like 50 proteins, it's multiple RNAs, and they all work together as a translating system between DNA and amino acids. So the current summary of abiogenesis, here's the scenario that I put together after reading multiple papers. And this is it's kind of funny. So between the edges of a hydrothermal vent, or between the edges of hydrothermal vents, ocean water, and a volcano rocky pore formed nature's own chemical laboratory. Each pore offered a unique microenvironment, stockpiling organic molecules. Lipid components delivered by meteorites crashing into Earth provided the raw materials for primitive membranes. In another location, lightning strikes while UV light radiated shallow pools, driving critical reactions. Meteorites crash near these pools, delivering essential elements and providing energy for RNA synthesis. Clay surfaces acted as molecular assembly lines, aligning and concentrating molecules in the pore of a rock, while magnetic minerals and crystals aligned with them into just the right configurations. This is to overcome the chirality problem. Furthermore, relentless wet-dry cycles in these pools allow them to link into more complex forms. Eventually, RNA migrated from the shallow pools to the hydrothermal vents. Tiny lipid bubbles emerged and encapsulated the RNA. And encapsulating RNA emerged while being able to hold the contents and transfer materials from inside and outside the cell without breaking down. It began migrating between these environments, scavenging components and energy wherever it could. Through this interplay of hydrothermal vents, meteorites, lightning, UV light, and wet and dry cycles, the beginning of the first cell was born.
Well, that's the craziest
Wow.
story I've ever heard in my life about anything. That's amazing.
I
Yeah,
picture
and
this as a
complex.
bedtime story that nerds tell their kids when they're really small.
Crazy. And this is why anybody that I'm talking to that believes this could happen, I'm like, tell me what environment this formed in. Because for each different molecule of the most primitive life form, they would need completely different conditions, completely different pHs, temperatures, environments, amounts of water and organics and different types of organics. It gets so crazy. So
Well,
for each
I've
one, they
had
have
my
pH.
sorry, Tay. I've had my mental hand up since line 3. What's with these lipid components, and how did their environment differ any from Earth's wherever they were created and then blasted apart and delivered via vacuum for God knows how many years in a vacuum and absolute cold, being on approach to Earth, being hit with 10,000 Kelvin solar winds, and then surviving the friction heat up on its way through the atmosphere to actually hit on the ground and survive that as well. No, those lipid components came via
Yeah. So I
that
was
way.
reading into the
Right.
papers on how lipid synthesis would have happened and how the membrane would have formed for early life. And they invoke meteorites contributing the necessary molecules for lipids to form and for the membranes to form. So
Now you talk about
I don't
faith.
know.
This
Yeah,
is faith right here.
a lot of it. Yeah, you're right.
This is worship. Holy moly.
That's the
You're
craziest conspiracy I've ever heard in my life. Like, and I've heard a bunch.
It really is. It's really bad. And if you actually go read some of these papers for each of the different components of the cell, you'll be just as shocked as I am. And I don't even think that I'm giving it its due diligence with how crazy it is and how many different scenarios and conditions and environments you would need to form each of these different components. It's crazy. All right. So I was trying to make some type of charts and bullet points of what all has been invoked for the most recent papers on abiogenesis to form different parts of the cell. So we have wet and dry cycles, as I mentioned earlier, lightning, UV light, clay, comets and meteorites, hydrothermal vents, shallow pools for the RNA, porous rocks with subsections of microenvironments having different pHs, and crystals to try to figure out the chirality problem. All right, so for the Miller-Urey clay, that's required for the formation of RNA oligomers, a role in biogenesis, RNA polymerization, and vesicle assembly. An environment would be in a tide pool or volcanic ash areas. So RNA is not in the hydrothermal vents. That would have to be in shallow pools. And the energy for it would be from UV light and literally, I'm not kidding you, with comet and meteorite impact, with asteroids impacting the Earth to have the energy to form some of the RNA bonds. I can cite that paper if you would like. It was very shocking for me to read that.
That is mystifying.
It is. I know, it's absolutely wild. And then pyrite for pyrophosphate synthesis, transamination, phosphorylation, energy generation, and this would be in a hydrothermal vent environment. The iron sulfur minerals for amino acid and nucleotide synthesis and redox reaction and metabolic catalysis. And this would happen near hydrothermal vents or in hydrothermal vents. And then the borate minerals for sugar preservation during formose reactions, ribose stabilization, and in evaporative environments, like maybe near a tide pool. And I'm telling you, this chart could take up the entire screen with all the different components, environments, and conditions needed for each different molecule. So if y'all
In the
remember,
correct order, too.
yeah, exactly, in the correct proportions. And by the way, if these were all in one condition, in one environment, they would cross-react. So they had to be made in different places, different locations, and different conditions. So I don't know if I want to go back up, but if you remember from earlier, there's 4 basic types of molecules for life. They have the nucleotides for RNA and DNA. They have the sugars for carbohydrates. All right, and then they have lipids for the membrane, and then they have amino acids for the proteins. So all of those form in different conditions with different like starting materials. Like if you read papers on how each of those form, they have different starting materials, different conditions. Even whenever they try to take something like formose and try to make 2 things from it, those 2 things that were formed in different pHs and temperatures. So there was like a paper on how there was like a starting material that could then react with different materials to make nucleobases and amino acids. But for each of those different reactions, it would have to be in different temperatures and pHs and conditions. And it cannot happen in a one-pot reaction because they would cross-react. So I just want you to understand that in the current like state of abiogenesis research, there's so many different sub-compartmentalized experiments that are needed and different conditions that are needed to form each of these different biomolecules. And that's the hard part. Is how you're going to get all these happening and then get them to come together to form life without cross-reacting and to continue to happen, because you have to continue to make amino acids and continue to make nucleotides and they have to continue to come together somehow without cross-reacting to form life. And it would have, I can't imagine how many different trials that you would need for it to go right and to get
At
all
the
the
correct saturation levels, too, of course.
yes,
Yes.
and the correct pHs, the correct temperatures. It's really crazy. So for DNA synthesis, there is some incompatibilities like with DNA synthesis, and this was like giving it the benefit of the doubt too. Energy source is UV radiation, heat, and it's around 55 to 85 degrees. And DNA and RNA are very similar. So because they're both made of nucleotides. So the DNA synthesis is prone to hydrolysis, and so is RNA. That's why you need the wet and dry cycles. It's paradoxical too because you would need UV radiation for RNA synthesis. And in one of their papers, they showed that UV radiation would give you energy for RNA synthesis. However, if it's exposed to UV light too long, it actually breaks down. And this is happening in the ponds, yeah, in the tide pools. So very paradoxical. And then these are stable at 55 to 85 degrees. However, protein synthesis requires temperatures greater than 100 degrees. So this is incompatible right here. 55 to 85 degrees for DNA RNA synthesis, but protein synthesis needs greater than 100 degrees. Isn't that
So
crazy?
it's 55 to 85 for the first two columns. For what length of time? Minimum.
Excuse me, I'm sorry, my throat. Okay, so it depends, like, you're asking, like, for what length of time would it take to form the DNA and RNA bonds? I mean,
Yeah,
I guess
in order to synthesize a piece of RNA or even a
yeah,
piece of DNA, for God, holy moly. The length of time, that'd take hours and hours, right?
I think if you have, like, all the perfect conditions with the activated nucleotides, then that could happen relatively quickly, but
Could you
you just
quantify that?
happen in nature. I know, right? Like, one of the problems is the activated nucleotides on the DNA and RNA, they cross-react heavily with the environment. That's how they can actually, react so easily with one another. And then so you'd have to have those very isolated and stable, not cross-reacting with the environment in 55 to 85 degrees Celsius, whereas protein synthesis, you need over 100 degrees Celsius. And actually, there's something called the Maillard reaction, which I go over later, whereas when you have these together in just an open environment, like RNA and proteins, they will actually, RNA and amino acids will cross-react in the Miller reaction. Yeah. I think I'm pronouncing that correctly. But basically, if you have these together in an open pot reaction, they will actually cross-react. But they couldn't have happened in the same location because they need different temperatures and different.
I'm working on a whole other angle there. So if you could quantify what I'm looking for, like for a correct start to finish DNA synthesis moment event to occur, what kind of time frame you're looking at?
I'm not sure. It depends. How many nucleotides you
Just
have.
a bigger window. Where do you think it would lie?
How long for the nucleotides to come together and form a bond?
Even just for a chunk of RNA to go from random chemicals sitting around in a pool with the correct
Oh my
85
God.
degrees and then
Oh,
boom, all of a sudden now it's a piece of RNA.
all right. So that's a big problem because we don't just start with like the basic chemicals and then have RNA or DNA form. What they do is they try to form the nucleobases, then they try to figure out how to get the sugar backbone on there. Those are each individual experiments. And then how to get activated with activated phosphates. That's one experiment in and of itself. And then
Okay, hold on. Then let's flatline those variables and say it's a scenario where all of those came in like Uber Eats delivered by your favorite meteor, your favorite asteroid, right next to those pools. And blammo, there was some shrapnel and they mixed together. And instantaneously, there was all of the abundance necessary to create RNA, to synthesize RNA. It's right there. How long would that process take at 55 to 85 degrees C?
I would say for like a short strand of RNA, I would
Sure.
say maybe a few minutes. But this is the thing is that
Minutes.
that's not plausible because even what they claim comes in on the nucleotides is only the nucleobase. So that doesn't have everything like the sugar backbone, which is a huge monster to tackle that they've been trying to tackle. Getting the ribose is absolutely amazing. The ribose sugar is amazing. And then also activated phosphates. So this is what I'm saying. Every single part of the scenario is broken down into individual experiments. There's not one cohesive experiment where they just start with the chemicals and then form activated nucleotides that bond RNAs. They'll start with either nucleobases and then, try to add a phosphate on there or, just individual steps to process need like different conditions and starting materials. So it's not like they can even form your scenario. Like they haven't demonstrated that to even occur. So even individual steps to even make DNA or RNA, those are completely different experiments and stuff.
I think in the back of their minds, they do understand that critical attrition is the natural process, is the natural tendency
Right.
in nature. There's no natural tendency to assemble. Quite the opposite.
Exactly. Yeah.
It's about decay.
Mhm. Well, this is funny too. So they have a quote, a requirement for UV light to generate biological building blocks would rule out deep sea environments like deep sea hydrothermal vents.
Yep.
And then another quote, to get one's electrons by UV photons at the cost of exposing a tender, unprotected neonatal metabolism to a notoriously destructive radiation, we would likely offer a much better deal, hydrothermal vents. So right here at the top, they're saying we would rule out like deep sea hydrothermal vents, and then this one, you would need hydrothermal vents. So it's like for different parts of the reaction, you just need completely different conditions, and the state of the research is a mess. So it's kind of hard to like put it in simple terms and just make like a cohesive explanation of the state of the research because it is all over the place. They're doing like little experiments for tiny processes, but having a cohesive picture is not there. And understandably it's not because you need such different conditions for each step of the way. And then right here, how long RNA versus protein survive in different environments. So aqueous at 77 degrees. All right, so this is the RNA and protein. So RNA would break down very quickly in aqueous environments at 77 degrees. Proteins would last about, what is this? Estimated half-life in days. All right, so 20 days max. And then let's see. So this is the 158 degrees, like they talk about the protein synthesis happening. Both of them would break down within days. And then in dry 77 degree Fahrenheit, you could have RNA last, or the estimated half-life of 40 days, and then proteins 80. But this is, it can't be continuously dry because remember, to get the reactions to form longer molecules, you have to have the wet environment to have them come into contact with one another. But then it has to have a dry environment, so they don't undergo hydrolysis. That's why you need these wet and dry cycles. And then dry at 158 degrees, the half-life is very short for both. So yeah, there's just some incompatibilities with the different environmental conditions for DNA and RNA synthesis and proteins, like with the temperatures here. All right, and then the Maillard reaction. Yeah, so here's the compatibility paradox. So amino acids can react with sugars like ribose, which is a component of RNA, in the Maillard type reaction, which could deplete the available ribose for RNA synthesis. So if you have the amino acids and RNA in the open environment, the Maillard reaction could occur, making them unusable for life. However, RNA and amino acids must coexist for life to form. You have to at some point have RNA and amino acids, which form proteins, come together to form the transcription translation system, which would be needed even for the simplest life forms that evolved to become us in their paradigm. So you have to have the proteins which make the DNA, and then the DNA which has the instructions for proteins, both of which are made up of DNA is made up of nucleotides, proteins are made up of amino acids. They had to come together at some point to make this happen. But if they're open in the open environment without the subcellular compartmentalization as we see in cells, they could just cross-react in the Maillard reaction. So somehow they formed in different environments yet came together and formed a cell. All right. So let's see. So here's another condition where they could be incompatible. In mixed prebiotic environments, amino acids and nucleotides could interfere with each other's polymerization. So basic amino acids such as lysine, arginine, and histidine significantly suppress RNA polymerization by competing with nucleotide binding sites. This competition can lead to mixed or undesired polymers, reducing the formation of RNA. However, RNA and amino acids must have coexisted to form life. All right, so now we're getting into a few of the paradoxes. The translation paradox in magnesium. So tRNA folding requires 1 to 5 millimoles of magnesium for optimal folding. And then So amino acid chelated magnesium of around 15 millimeters total concentration increases RNA folding and stability while maintaining around 2 millimoles of free magnesium. And then for RNA bonding, so this requires lower magnesium concentrations of around 0.5 to 1 millimoles free magnesium in the cells. And nucleotide synthesis, magnesium concentrations over 2 millimoles suppress nucleotide incorporation into DNA. So right here for nucleotide synthesis, you cannot have too high of magnesium present, but you require over 2 millimoles equal to or greater than 2 millimoles for the RNA folding and stability. So in the cell, we overcome these problems by having things, having things separated and having different parts of the cell with the different concentrations that are needed for, life to continue. But out in the open environment, it's like you wouldn't have any of this compartmentalization, and you could just have these cross reactions and incompatible conditions occur. That was just an example with the magnesium. And then here's the phosphate problem that I mentioned earlier. So phosphate was required for the DNA backbone to be created. However, phosphorus-containing minerals on the early Earth, such as calcium phosphate, were highly insoluble, making them biologically unavailable. So it was funny because whenever this problem was brought up, NASA tried to overcome it with this new arsenic life form, whereas arsenic could take the backbone of phosphorus. Arsenic could replace the backbone of phosphorus in this early life form. So they were proposing that they found a life form in this lake, the Mono Lake in California, that had an arsenic backbone instead of a phosphorus backbone, and this could kind of overcome the phosphorus problem of how phosphorus was unavailable bioorganically. So it was basically bound up in like calcium phosphate and insoluble. So they were claiming, this new arsenic life form, which was later debunked by other scientists. So NASA falsely claimed that life forms could get around the need for phosphate by using arsenic as a DNA backbone, but scientists have later called out NASA. So here's a paper right here. Scientists say NASA's new arsenic life form was untrue. So they found that NASA's research on this was not true, and that there was some fishy stuff that they were doing within the research to show that arsenic could replace phosphorus in the DNA backbone. So it was kind of funny to see NASA get called out by the biological community. It was the first time that I saw that happen. All right. A few more paradoxes. We got the water paradox. So water is commonly viewed as essential for life. So are biopolymers like RNA, DNA, and proteins. However, these biopolymers are corroded by water. For example, hydrolytic deamination of DNA and RNA nucleobases is rapid and irreversible, as is the base catalyzed cleavage of RNA in water. So water is required for the mixing of the different amino acids per se to get them next to each other to form proteins. However, whenever the protein bonding actually has to occur, like this has to happen in like water-free environments or low water environments, so that you don't have hydrolysis occurring, so they don't break apart. So it's funny because you need water, but then again, if water is present, hydrolysis will occur and things will just break down, like the hydrolytic deamination of DNA and RNA.
I remember that the first class in high school on the subject where they mentioned water is the universal solvent, is it not?
Yes, and water would be required for that's why they're looking for water on other planets, but actually, like, available water on the we have not, even the mainstream claims that we have not found water on the surface of other planets. It's either ice and, like, liquid water would be, like, under the surface, or, just, like, the planet's covered in ice, essentially. We don't see liquid water on the surface of other planets. That makes Earth unique. That's another thing that makes it unique, and it would be required for life.
Yeah, this is getting into comical now.
Sorry, go ahead.
I was just mentioning how this part of the plot is getting into the comical world.
Exactly. Yeah, but it's just another paradox, like with the origin of life, even in their own mainstream model. Like, even assuming their mainstream model, there's a huge problem here. So RNA requires water to function, but RNA cannot emerge in water and does not persist in water without repair. So, and other things like, protein formation can happen. This is why you need the wet and dry cycles. All right, so the energy paradox, energy is needed for life. However, raw energy without proper ways to process it will cause destruction. For example, trying to charge your phone on a charcoal grill. You're not like, it's energy, but it's not the energy that's usable for life, because energy that doesn't have a way to process it, like, is going to be deleterious. Or putting a Tesla charger in a gasoline intake hole. So without the proper ways to use this energy, it is destructive. Kind of like the UV light paradox I mentioned earlier. Like, you need energy for the RNA bonds to form, and they invoke UV light, but like RNA exposed to UV light for too long will break it down. And DNA exposed to UV light for too long will break it down. So yeah, the UV paradox was next. Yeah, crucial for certain bonds to happen, but it will break down and destroy the very molecules it helps create. It breaks down DNA and forms pyrimidine dimers and photodegrades organic phosphorus compounds. But earlier, as we saw, they were invoking it as the energy source for RNA bonds. So here's another paradox, the hydrothermal vent paradox. Hydrothermal vents are proposed for protein synthesis, yet the high pH of alkaline vents strangle out carbon life needs and is destructive to RNA. So there's a little paradox here. So you need the pH of alkaline vents for protein formation, but it'll strangle out the carbon life needs and can be destructive to RNA. A study published in Nature Geoscience revealed that hydrothermal vent conditions dissolved free organic carbon compounds to carbon dioxide. They wrote that even the most resilient organic molecules eventually broke down as they circulate through the vent systems. So they actually find, like, as they're circulating through the vent systems, it will break them down. But remember, we need these alkaline environments and high temperatures for protein bond formation. Because since you don't have enzymes to overcome the activation energy, you're going to need these certain conditions to get proteins to bond. All right, the heat paradox. So heat is a catalyst and will accelerate chemical reactions, providing energy for forming complex organic molecules. But heat is also a destructive force as it breaks down the delicate biological molecules like RNA and proteins. So you need heat, but whenever you have it, it will also break down biological molecules. And then the natural selection paradox. So researchers appeal to natural selection to get complex machinery, but you can't have natural selection occur until you get self-replication, which requires complex machinery. So this is a funny one. Yeah, appealing to natural selection to get complex machinery, but you can't have natural selection until you have a self-replicating cell, and it can, like, have different life forms you can select from. You cannot have that until you have complex machinery, because you need complex machinery for cell replication. So, funny side note too. The time paradox is another one. So complex organic molecules require time to accumulate and become useful through random chemical processes. However, time is the very thing that will degrade organic materials. So over time, RNAs and proteins in the environment will break down. So they're literally racing against the clock. So people think, oh, with enough time, like, you can get basically any organic molecules to form, and proteins and RNAs and all these things in the right sequence. But time breaks down these molecules. Like we saw the half-lives earlier. So you're racing against the clock. And then DNA protein paradox. So modern cells require proteins to replicate DNA and RNA, but proteins themselves are encoded by these genetic materials. So this creates a circular dependency where the components necessary for life rely on each other to exist, complicating the understanding of how life could begin from simple organic compounds. So yeah, you need proteins to form DNA, like right here in the picture to the right. Proteins used to produce DNA. DNA is used to produce proteins. So DNA has the code for the proteins. So you also need proteins to produce RNA. Proteins will basically help RNA replicate. And then you have RNA The primers necessary for replication of DNA, and then RNA as rRNA and tRNA is used to produce proteins. All right, so proteins used for translation of RNA and used for transcription. Sorry, DNA is used for transcription to RNA and then used for translation to protein. So it goes from DNA to RNA protein through transcription translation. So these all codepend on one another. That is the point. DNA, RNA, and proteins, they all need one another in order to have, basic life in a cell. You need to have proteins, you need to have DNA, which gives a code for it, and they all rely on one another to be produced. So it's very circular. Like, what came first, the chicken or the egg? So it's
Yes. The answer is yes.
That's so funny. So the translation system requires a highly evolved set of RNAs, proteins, and enzymes for high fidelity. However, these proteins are formed via the translation system. Another chicken and the egg problem. I think tRNA is going to be coming up soon, Dan, if you're here, but I have a couple more slides before we get there. So Eigen's paradox, we cannot have long strands of DNA unless it can repair itself. And it can't repair itself without long strands of DNA. Okay, so this was posted in Nature. So he posed the paradox that without error-correcting enzymes, the length of a nucleic acid would be limited because in larger molecules, mutations would destroy the information content of subsequent generations. But this maximum size or error threshold was too small to encode the error-correcting enzyme. Eigen's paradox still challenges theoretical biologists today. It's funny calling them theoretical biologists, because we can't actually replicate life forming in the lab. But we can do tiny little steps to process. Anyway, so this Eigen's paradox can still challenges biologists today. And so you basically need a long strand of DNA to produce mutation correcting machinery. However, to make that mutation correcting machinery, you need to have long strands of DNA because it is a very complex thing. Dan, off the top of your head, do you remember any of I know DNA polymerase will correct mutations. Do you know of any other proteins and molecules off the top of your head that go back and correct it? Like whenever there are mutations? I think helicase.
For translation, there's also correction in the ribosome, but it pauses while it corrects. It doesn't walk backwards. It has to pause. It pauses while it's waiting for correction. But we can talk about that a little bit more when we go over the aminoacyltransferase.
Yeah. So they have machinery that will actually go back and correct if there are mutations. And this is some questions I've been talking to evolutionists about. Like if you have these new genes form or these insertions to make new genes, how does that continue? Because whenever you have insertions of DNA, it'll actually prevent correct transcription of the DNA and replication of the DNA. So anyway, the cell has correcting mechanisms to make sure we don't have too many mutations. If there are mutations, it goes back and repairs itself. So the membrane paradox is another one. So this is the membrane formation paradox. It acknowledges cell membranes can only be synthesized from pre-existing membranes. As an extension, the organelle formation paradox states that membrane encapsulated organelles can only be produced from pre-existing organelles. And the protein phospholipid paradox demonstrates that protein composition in organelles is dictated by the organelles' phospholipid composition, but the phospholipid composition is dictated by the protein composition. So there's a lot of circular things to here. So how did these things ever start off? Can we replicate the beginning of these processes? This is
That's hilarious.
Definitely. All right. So there's a lot of circular synthesis paradoxes as well. So for example, you need 6 ATPs to make 1 ATP. You need NAD to make NAD. You need tetrahydrofolate to make tetrahydrofolate. You need coenzyme A to make coenzyme A, and FAD to make FAD. So this is the citric acid cycle over here on the right that happens within the cell. And you can see a lot of the things that are produced are also required. So it's just a lot of circular, how would you call it? Circular synthesis, where you need the thing to make the thing. So how did the thing ever come about? Anyway, it's funny. So the ribozyme paradox. Okay, so this is kind of important because it actually is like the leading theory. The RNA first hypothesis is the most cited theory. So there is a paradox with the ribosomes. Let's see, one second. Okay. For a ribozyme, so they basically invoke a ribozyme, which is a self-replicating. So they basically took a RNA polymerase and tweaked it up to make it self-replicating, and then they proposed that this could have been produced through nucleotides forming RNAs. And then basically they say these nucleotides could have formed a ribozyme, which could self-replicate and then start off the replication process. All right, but whenever they did this, they actually just tweaked up an RNA polymerase and took a protein out of an already living cell and mutated it to do this like self-replicating process. But that didn't happen like naturally. Okay, so for a ribozyme to be functional, it must be stably folded into a complex structure, much like a modern-day enzyme. However, in the absence of a set-aside genome, this same ribozyme would also need to serve as a template for its own replication. So the templates cannot have complex stable folding if they are to be accessible to free nucleotides and an RNA primer. Clearly, the same RNA cannot meet these two mutually exclusive requirements simultaneously. In the RNA world hypothesis, ribozymes necessarily must have dual roles. They must have the catalytic activity to replicate while simultaneously being able to act as a template for replication. Catalytic activity would require stable folding into complex structures, whereas its role as a template requires unstable and relaxed folding. So if it was to like self-replicate, you'd basically need two of them, because you need the ribozyme, which has catalytic activity, but you'd also need the template and the instructions for that self-replicating enzyme. So like it has to be folded to have the catalytic properties, but unfolded to like grab the sequence from it to replicate it. So there's a lot of these like circular just issues that they're having to overcome. So this is a funny paper called The RNA World Hypothesis, the worst theory of early evolution of life, except for all the others. So this is funny. And this paper talks about how RNA world is not a good theory because RNA is like so unstable. So this is talking about how, coming against people who say RNA first and how they're still kind of trying to come to a general consensus of even what biopolymer came first to produce life. So and they do acknowledge here in this paper, except for all the others. So it's kind of funny. There's problems with every theory of abiogenesis. All right, so here's Dr. John Sutherland, who has made significant quote unquote advancements in origin of life research, and he concedes that there is a lack of progress in explaining a naturalistic origin of life. So this is a chart that was in one of his papers. So degree of aliveness is on the y-axis. Then you got the time or system complexity. So at the start, we have inherently favored prebiotic chemistry, energy dissipation, systems innovation, optimization of major innovation, necessity, contingency, boundary, and evolutionary innovation. So see how it had to be fully alive before you have evolution kick in? So you can't invoke any evolutionary process until you have a fully alive cell. And it's funny, like whenever they talk about natural selection, oh yeah, like this and that, they could have just like chose the right things. No, chemicals are not alive. They just react. They have no outcome or goals. They're just haphazardly reacting in the reactions they naturally do. They don't have an end in mind. They're not choosing the reaction. They're not choosing particularly left-handed amino acids. Like, hey guys, we need just the left-handed amino acids to make a protein. They are not alive. Okay, so all these processes I'm going over are completely undirected. They just haphazardly occur. And shall I mention that they haphazardly occur away from life normally? Normally, biopolymers break down. They do not build up in nature. So you're having to overcome the natural way of how we see things operate at the chemical level. So he argues the current state of the field is right here at the bottom, bottom of the screen. Look at the little blue box to the left. The current state of the field. So this is an abiogenesis researcher talking about the current state of the field being here. where they're trying to figure out the favored prebiotic chemistry. Now, the next step in this paper is saying the energy dissipation, exploration of macromolecular sequence and composition space. And he's saying the current state of the field is here, but it's funny because I added this line and I say that I argue that it's like way back here because honestly, the more we learn about the cell, the more questions we have than answers on how it could have formed. All right. So over 100 years after abiogenesis was proposed, scientists have yet to successfully create a functional protein from inorganic or basic molecules under prebiotic conditions. That's really important. Scientists have yet to also create a functional RNA molecule or RNA strand from inorganic molecules under prebiotic conditions. So we do not have like a functioning RNA or a functional protein that has ever been formed from basic molecules. Okay, so a lot of people would lead you to think that we have reached these milestones, but no, 100 years of abiogenesis research has been occurring, and yes, we haven't even reached these basic milestones of forming just the basic polymers, just like a basic, RNA strand or protein strand, just from chemicals, like
Exactly. Like you said, they have no natural tendency to assemble.
Right. So, like, for example, if they took the Miller-Urey experiment where they struck like the methane and ammonia, like with lightning and stuff, and then they formed that tar that had like 2% amino acids, like then like they need to take those 2% of amino acids and get them to come and form like a protein that's functional in reasonable conditions that are undirected. Okay. So that would be something that would need to be shown, that these basic molecules can then form a functional protein. But yeah, we have yet to show that occur. Same thing for the RNA. And the RNA is way harder, getting that ribose, the sugar backbone, and the activated nucleotide, getting those to form like an RNA, much less a functional one, has yet to be shown. All right, so they're acknowledging here, John Sutherland, that we are not yet close to achieving this end. I guess I'll read this quote real quick. Understanding how life on Earth may have originated is a major goal of origins of life chemistry. To proceed from simple feedstock molecules and energy sources to a living system requires extensive synthesis and coordinated assembly to occur over numerous steps, which are governed by only by environmental factors and inherent chemical reactivity. Demonstrating such a process in the laboratory would show how life can start from the inanimate. If the starting materials were irrefutably primordial and the end result happened to bear any uncanny resemblance to extant biology for what turned out to be purely chemical reasons, albeit elegantly subtle ones, then it could be a recapitulation of the way that natural life originated. Yet we are not close to achieving this end, but recent results suggest that we may have nearly finished the first phase, the beginning. Emphasis added, the first phase. The prebiotic synthesis of building blocks to which we have devoted so much of our time only correspond to a small increase in the complexity of the system and to no increase in its aliveness. A humbling thought, he states. So yeah, if you actually talk to origin of life researchers, they will acknowledge the lack of progress in the field. Like they will, if they are honest, will acknowledge that we are not even close to achieving this end. Okay, we have and they've been researching it for 100 years. All right, the next segment is separate from the last one. It's a positive case for intelligent design within the cell. So indications of an intelligent designer. All right, so we can look at things in our world and see what is intelligently designed and what isn't by some of these key indicators. Like if we find a computer or a car on the side of the road, there are key indicators to suggest that, hey, this thing didn't just pop up out of existence. This thing was intelligently designed with a purpose and with functionality. All right, so here's some indicators of something that is intelligently designed. So it has complex and specified information and organized meaningful complexity. It has irreducible complexity, all parts essential for the function or the fact that you need multiple parts for it to function. Okay, when people say irreducible complexity, please be careful when you say that, because other like pro-abiogenesis individuals will say, oh well, look, we can remove this like insignificant like piece of the flagellum and look, it still rotates, it still functions. Okay, so that's why I don't like using the word irreducible complexity, because they can say like, oh, this like insignificant side thing was removed and like it still functions. So don't use that. Whenever you use it, emphasize the fact that you need more than one part for it to function. Okay. So you need multiple parts for it to function. It couldn't just happen in a bottom-up manner, because you need multiple parts for it to function. All right, purposeful arrangement. Features designed for specific goals. Fine-tuning, precise calibration of parameters, information-rich features, detailed intelligence, associated information, specified complexity, complex non-random patterns matching specifications, adaptation of means to an end, parts working together for a clear purpose, and creation of new information. So novel function information, suggesting intelligence. So on the right, this is like, this is in like developmental biology and all like the intricate integrated systems, correlating and collaborating with one another. So you got the mesoderm and the ectoderm all sending signals to one another. And this is for development of like a cell or for an organism. Dan, is there anything you want to mention on this right image? I don't know. I should have like wrote some more like writing for this, but I'm sure you can recognize some of the Wnt8 and the let's see.
Yeah, I
escape.
mean it's been a little while since I looked at, especially the stuff at the top. Like these are like second messenger pathways and things in development. You see how you see mesoderm and endoderm at the top?
Yeah.
It has to do with basically telling cells like whether like this all involves like development, right? So telling a cell like what it's going to become. I mean, can you imagine? Like, look how interconnected all this is, like, and helps in like organization of tissues and body structures and stuff.
Yeah. And it's crazy, like this reminds me of like a computer software system or something like that. You know, like a computer chip, and how they're all like corresponding with one another, and yeah.
We got a computer chip slide coming up. That's it. There's another
Yeah.
one.
It really mimics it. So we know that computers are intelligently designed, and they're little softwares and their chip. I'm not like an engineer by all means, but like, yeah, this reminds me of like computer pathways and stuff, even though I'm not an expert in that topic. But yeah, they are definitely intelligently designed as well. And when we see things like this happen within the cell and these pathways that are interconnected, and like you got this specified complexity and fine-tuning and purposeful arrangement of things, like this is definitely an indicator of intelligent design within the cell. All right, so integrated systems and protein synthesis, the system of DNA, mRNA, and tRNA and ribosomes work together in a tightly integrated network. Each component is dependent on the others for its function, or else death or disease could occur. So tRNA, how does DNA get translated into a protein? So this is very intricate, as I mentioned earlier. Daniel, did you want to go talk about the tRNA, some of its requirements to
Yeah,
make
this slide just real quick. This is just basically showing the translator tool. If you want to kind of manually translate between RNA and DNA and RNA, this is the codon translating wheel.
Yeah,
And
so
I'm basically just saying that there's two specific chemical languages, and they function differently in two different important ways. There's the different spellings, the orthography, and then it's a different system of units, the morphology, right? So DNA is like chemically different than RNA, and then they are arranged differently for their meanings. Okay,
yeah, you got
we can go to the next slide.
you got two language systems. Like you got the language system of DNA with the nucleotides, and the language system of proteins, which are amino acids. And then so 3 nucleotides will code for 1 amino acid. So this is called codons. So 2 different language systems using different spelling and morphology. They have to be matched up. What does this? Okay, well, the tRNA matches them up. So there are 64 possible codons from mRNA, which must be matched to 20 different amino acids. And then so here is the tRNA right here. Did you want to say anything on this one, Dan?
Oh yeah, for sure. So most importantly, like what we're going to talk about is a process called translation. And if you look at the picture on the right side, you'll see the big red bubble. That's representing the ribosome. And the ribosome is, we'll go in detail on another slide, but it's made of a bunch of different components, and it has different positions where it slides along the RNA and then uses a tRNA to build each peptide, each amino acid to build proteins. Okay, so um, its role is to act like a translator between these two different languages. And there is a specific tRNA, which is, if you see the purple strings, that are
Yeah.
going into the red circle,
Mm-hmm.
each one of those have to be made with its little anticodon sequence. And that anticodon sequence is what binds it to the RNA, but it has to be activated with the little green balls on the top. See all the way to the right, there's a a proline green ball on the purple string,
Yeah.
and it needs that to begin. That's the first codon.
Right. And then I
Okay,
think, yeah,
perfect.
we
Here
have
we
two
go.
more. Yeah.
Thank you. So zooming in on that purple string, we can see this is the structure of a tRNA. The left-hand picture is when it's just laid flat to try to demonstrate the difference between the D arm and the T arm, and the anticodon on the bottom and all that. But this is really cool here because I told you that the purple string, the tRNA has to be activated, right? It needs a peptide bound to it, which we'll get to next. Oh, and it happens at the acceptor stem, which is on the end. I really
Yeah.
love the picture in the middle. Look how things really are. Like, we try to demonstrate them in a simple way so we can study them and describe them, but everything is folded up in this complex 3D geometry form. Next slide. So here is the process of tRNA charging, which I think is really cool. So a preformed protein, I really want to emphasize this process is required to make every single known protein. Okay.
Mm-hmm.
To
And protein.
initiate the process, to even get ready to think about translating something, you need a preformed protein called amino-acyl tRNA synthetase
Mm-hmm.
on the bottom left. Okay, on the little green writing. Okay, and you see that is the protein which is gonna activate each tRNA to its unique and correct amino acids, which is 20 different ones. And you need 20 different proteins and RNAs needed for just one protein to be synthesized. Okay, so
Isn't that
um,
crazy?
yeah, and here on the bottom left, there's actually the folded form of the amino-acyl tRNA synthetase.
Yeah, that's crazy.
Next slide. Perfect.
You got the Taylor?
Yeah.
Protein
So
synthesis happens at the site of the ribosome. Ribosomes are molecular machines made of two very large RNAs. Okay, so this is a little bit more details about the ribosome itself. Remember on the picture a few slides back, we had the red ball with the purple strings going through it. And this ribosome has three different sites, right? The A site, I always say like activation site to help remember that's where it starts. And then there's the P site where it starts adding on to the one in front of it. I think of P for peptide because that's where we're building peptides, right? Where the amino acids from the TR adds on. It's the
yeah,
elongation site. Then you have
right.
the exit site, the E. This is where like the tRNAs need to be able to pop out of the ribosome, and they're going to get activated again and be reused. But and
Mm-hmm.
then it's hard to see. You have to zoom way in. But if you look at the picture at the top, you can
Yeah.
see the ribosome going through this little cycle, and it builds a little chain of peptides on the top of it.
Mm-hmm.
And then all the way to the right, just some details of the ribosome. You can go to the next slide. There's should be a better ribosome slide.
Let me see. Okay.
This is
Oh,
basically like if you thought like oh, that's it to what I was describing. Like, no, there's even more details about each one of these components that we're referring to, and they all require like some other component or protein to interact with them and hold them in the position they need to be in.
Crazy.
If you wanted to like learn all about like protein synthesis, you could take a screenshot of this right here. This is like a page out of first aid for just kind of what you need to know about protein synthesis.
Yeah, it's very complex. So the ribosomal components, the bacterial ribosome contains around 54 proteins. And remember, this is for one protein to form. And you need proteins to form DNA. So it's just, yeah, it's a lot of like things that are circular. I can carry it on from here, Dan.
Wait. I guess that's the one thing I knew. I think I took a slide out, but I was wanting to really emphasize that like you already talked about the definition of irreducible complexity. That you take one
Yeah.
piece out, the whole machine quits working.
Yeah,
and
this one.
the ribosome is great. And that if you remove one of the 50 essential proteins in a ribosome, the whole thing quits working. If you don't have the aminoacyl tRNA synthetase to activate the tRNAs, then the ribosome's useless.
Yep.
So it's just incredible, really.
Yeah, I know. This one in particular, yeah, very prone. If anything's removed, it's not going to function properly. And so here are animations, and I think I had some electron micrographs too, showing these things that aren't animations. I showed you the microtubules that, this is basically a kinesin transporting something within the cell, walking on the microtubules. And I showed you the microtubule elongation from the fluorescent microscopy that I view in live cells. And then, like, here's the flagellar rotor, and then this is DNA replication happening right here on the bottom left, an animation of DNA replication occurring.
These geometries are immaculate.
I
How
know.
can anybody dream that this randomly forms and ordinates itself like this, and that operates as a mechanism with the correct proportion and intent? Who's dreaming about that?
Well, what
I
about the
don't know.
lightning?
You can literally like make these different proteins fluorescent, so you can actually watch this occurring in live cells and watch their interdynamics. So it's amazing to look at the DNA replication occur. And then
Yeah,
here's the mitochondrial
I-
ATP synthetase right here, making ATP.
that's a whole other conversation. I feel like we could go on 10 slides just talking about ATP synthase. Because this rotor that's spinning is driven by a proton force, basically like an electrical charge, and then it twists and apparently is like squeezing phosphates onto ADP to make ATP, and we don't really understand how it works actually, because it really basically like on it gets stuck to the rotor better whenever it's ATP. And so we don't really know why it does the way it does. And it's really just a mystery, to be honest. It's pretty cool.
Yeah, it's amazing. These little machines, like, these are absolutely amazing, and they turn like this. I know for some of these, like, they rotate faster than a jet engine, and they have like almost 100% efficiency, unlike the engines that we create for, like, cars and stuff. They might have like higher efficiencies, like 80% or so, but this has like 100% efficiency. And it's absolutely amazing. You could like literally make 100 slide presentation on each one of these, and how amazing the machinery is. And we can't even come close to replicating it. And we can't even replicate it in the best conditions possible in the lab from scratch. So it is what it is. All right. Did you want to talk about this one then? The similarities in the ATP synthase and man-made motors. I think I made this slide though. So
Yeah, you definitely made this slide, but it's really awesome. Yeah.
oh, here's some pictures.
You can see like just these great pictures. Like, so I like the one at the top right with the little green ball and the blue gear at the bottom. Okay. It's called the rotor. The green ball, I think it gets the name from an electrical motor, and then you have like a spoke, and then the blue gear at the bottom. That's what's getting driven by the proton force of hydrogen ions across the mitochondrial membrane.
Yes.
If we need to talk about that, we can like make another slide on that for future references. But it's so cool how this thing is using torque generated by the proton gradient in order to like mechanically squeeze a molecule to add on phosphates. And then I'm sure most people have heard of ATP, right? It's like energy of the cell. You know, it's like what's needed for any biological process. It's like what helps every single one of your muscle cells move, you know?
The currency of
Um,
the cell, the energy
yeah.
currency of all living cells, yeah.
And
ATP.
heal.
Go ahead.
Oh, that's it. Does someone
You can
say
mute up.
something?
I can hear someone in the background. Thank you so
Well,
much
I
for
was
your
just
help.
saying that ATP also helps cells to heal.
Oh
Wow.
yeah, for sure. Every single one of your cells, for it to start doing something and making some process, or even just to stay alive, like it's using ATP.
Thank you so much for your contributions, Dan. I know you're working, and I just appreciate you for coming out and helping a little bit for these slides and talking about the
Oh, no,
slides. Thank you
this
so
is
much.
super cool. I could do this all day long. Thank y'all for being here and like paying, like, you know, contributing and listening and stuff.
Yeah, no problem. Thank you so much. All right, so here's something I have on this. So they have actually pictures of this ATP synthase right here, within the cell. So you can see pictures of it, and then just like an animation to kind of show what's happening and the rotors and everything, and the rotation, and how you have sodium coming in, and basically you have ADP plus phosphate to produce ATP, which is like the phosphates being used is like what's like the energy of the used as energy of the cell. So it's like the phosphate on the ATP being expended. And this is how ATP is made through this ATP synthase. All right, and there are papers still trying to unravel fully the mechanisms of ATP synthase. My biochemistry teacher in undergrad was talking about how amazing this is and how we're still trying to fully understand it. And I'm not sure if they're believe in intelligent design or not, but they still acknowledge the amazing intricacies of this process in ATP synthase. All right, so ATP synthase and man-made motors share key similarities. Both utilize rotational mechanisms with a central shaft rotating within a stationary housing, resembling the rotor and stator design in electric motors. They generate torque to perform work and are modular in structure and adaptable, such as how ATP synthase can operate in reverse. Additionally, they convert energy with remarkable efficiency. ATP synthase achieves a near 100% efficiency. So this was like the comparison between ATP synthase and man-made motors, but these actually have 100% efficiency, near 100% efficiency, whereas man-made motors do not. So it's funny how like in cars and other things of that nature, boats, cars, whatever, an engine, we actually like made these things and like use this design here for motors. And then whenever we looked inside the cell, we saw that they're utilizing the same exact mechanism. So it's like, okay, we intelligently designed a motor to work because the way that its design works. Okay, so this design works. Okay, then we look inside a cell and see the same design. So obviously it's there because it works too, but ours is intelligently designed, but we will assume that this wasn't intelligently designed. Like, it's crazy. Anyway, so yeah, these different components are required for it to work. All right, so in DNA, there's also evidence of intelligent design, such as complex specific information. So it's complex in that the different nucleotides A, T, C, and G have complexity, and it has specificity. So it actually codes for something. The nucleotides code for different protein sequences, amino acid sequences to make a protein. So people will say, okay, well, you'll have like a crystal is complex, like it'll have like repeating things, like repeating chemicals or whatever, but it's not coding for anything. It's not specific information. You can say a mountain's complex because if you look at all the rocks on it, it's a very complex thing to like map out all the rocks on a mountain, but it's not specific. They're not coding for anything. So that's what makes DNA unique. It is complex and it's specific, right? Specifically ordered. Information being a message or instruction with meaning. So it has information-rich features. It stores and transmits detailed genetic information. It has specified complexity. DNA's base pair patterns are complex and non-random, matching specifications for cellular functions. It has purposeful arrangements. DNA structures designed for storing and transmitting genetic information. Irreducible complexity. The replication process is irreducibly complex. You have to have that tRNA. You got to have it go to RNA, which forms, which then gives the instruction for protein. You got to have that tRNA there, which requires like 50 proteins present. So there are multiple things that are required for DNA replication and for transcription translation to occur. All right. And like the DNA polymerase is absolutely amazing too. That's one of the things on the last slides. Right here for the DNA replication. All right, and then fine-tuning precise nucleotide arrangements in DNA sequences produce functional proteins. All right, so this is some famous, I guess you can call them scientists, and their quotes about DNA. So Bill Gates, Microsoft founder, said DNA is like a computer program, but far, far more advanced than any software ever created. Richard Dawkins said the machine code of the genes is uncannily computer-like. Francis Collins, who ran the Human Genome Project, DNA is something like the hard drive on your computer containing programming. Craig Venter, life is a DNA software system containing digital information or digital code. So a lot of people are like pointing out the similarities between DNA and computer codes. All right, so evidence for intelligent design. So informational hierarchy in integrated systems. So just as intelligent agents designed integrated circuits that are precisely, I think someone's hot micing. Just want to let you know to meet up. It's all good. All right, so they're integrated circuits that are precisely engineered with billions of transistors. The cell has these circuits composed of intricate molecular components performing complex information processing tasks. So to the left is like a circuit board for a computer. Circuitry, informational hierarchy, integrated systems, and computer chips, and then also circuitry within the cells. This is amazing. This is like metabolism over here on the right. I can't zoom in to see all the little molecules, but it is very complex, and we're still adding to this today and fully understanding it. But you can see just like the integrated systems within a cell also match like the circuit boards within a computer. I'm not sure if Dan wanted to say anything or not.
Yeah,
but,
sorry, I was hot micing. I just wanted to. That was that slide I was referring to about the computer chip and stuff. And then I know I wish we could zoom in on the biochem like picture to the right, but it looks more complex than the computer chip in my opinion.
Oh
Anyways,
yeah, it has
you can keep
specific
going.
cycle, I believe, too. Yeah. All right, so computational architecture of the cell evidence for top-down design. So like for computers, like we design it top-down because we have like an end in mind. We know all these different functional parts that are needed to make the computer work. And same thing for a cell, like it has all these functional parts that are needed and integrated systems needed for it to work, even the simplest possible cell. So like I believe it's more reasonable to say that it was designed top down since it does need multiple systems like a computer. Like, can we conceive of a computer that could be like designed bottom up just by like little pieces of metal and then like coming together, like forming something that's useful, and then it actually needs this other thing like a motherboard
I think
needs
they called it an abacus.
I don't know. There's a slide I thought it had, but
That was a
there's
joke. Okay.
there's parts of a computer that require one another as well. But yeah. So the processing engine, like in a computer, you'll have like the gate, and then the implementation in a cell is like the amino acids. And then let's see. So like a latch, we got the primary main memory gate, and then the nucleotide is a basic element. Engineering gene circuit. There's a whole paper on this actually with the comparisons of a computer and the cell and how they have a lot of similarities. So yeah, the controlling path for the instruction, similar to a protein, proposes basic computational elements in living cells. So the complexity of the cell and its corresponding analogies to contemporary computing systems run deeper than simply DNA. The cell as a whole has also been viewed as a computational machine similar to a multi-processing cluster with a combination of centralized instruction set and distributed computing elements. So the channels of information transfer and transformation in the cell have direct analogies in communication theory and computational algorithms. So yeah, there's a lot of similarities. I think it was the CPU and the motherboard that rely on one another in a computer that like are needed together. But like I said, I'm not like an engineer or computer specialist whatsoever. But there are a lot of similarities within the cell and computer systems. Were you gonna say something else, Tom? All right. Okay, so is abiogenesis scientifically plausible? So a scientific explanation must be coherent and provide evidence to whatever phenomena it is trying to explain. So abiogenesis lacks this. It doesn't have like a coherent process. It doesn't have like a coherent model from start to finish. And it definitely doesn't have one that's testable. I even ask people that believe in abiogenesis, like, can you give me any sort of scenario or model as to how these chemicals could have formed biomolecules and how that could have led to anything that's like self-replicating that could lead to life. So they cannot break this down for me, like, because they understand that like the RNA, the proteins, they need different environments, and how they would have came together without cross-reacting and formed a cell and like had this transcription translation system form that's like it's amazing. They can't do it. They can't explain how this could have actually formed. Like they can't even conceive of it. Much less replicate it. And so if we're going to say that it's like plausible, I feel like we should have like some coherent model or idea of how it could have occurred, and have it be testable. And I've heard in return, like, okay, well, intelligent design doesn't have this. Intelligent design can't show like it model it out. But the thing is they're describing an explanation in the natural realm. We actually work in the natural realm. Okay, so if we work in this realm, then you have the advantage, and you should be able to describe how chemicals form life. If you're saying chemicals formed life, well, we can work with chemicals in the cell and see the nature of them and how they behave. Do they form life or do they move away from it? It seems like they actually move away from life, but they have to come up with some, crazy processes as to how they could form life. But they can't like come up with a coherent model or replicate it. So, I mean, any like hypothesis or thing that's being proposed as scientifically plausible should at least have a coherent explanation and be able to demonstrate it.
I want to see an 8-part Netflix series on it.
I don't even know if you got the scientists together, they couldn't even come up with how they would put it together. And then a lot of times they'll say, oh, well, we'll figure it out one day. Think about the 1900s, like where we're at now, where we're at in technology. Okay, well, within 100 years, we've also been researching abiogenesis, but we're going in reverse with the understanding of it. We're actually learning that the cell's more complex. We're getting further away from understanding how it could have occurred. And then saying that we'll figure it out one day in the future is just like kind of like an appeal to the future fallacy. And ironically, the more we learn, the more unreasonable abiogenesis becomes and seems to us. So is intelligent design a proper alternative? This is the big question, because it's like scientists won't acknowledge an intelligent designer because they say it's not scientific because it's not testable. Okay, well, honestly, yours isn't testable either. And you don't even have like a conceivable model as to how it could have like happened. And if there is one, please bring it forward. So Occam's razor refers to explanations with fewer abstract postulates. Intelligent design has one postulate, an intelligent designer. There is so many abstract postulates and scenarios that they have to like invoke for abiogenesis and that are not demonstratable. So this is what makes them like abstract, and they're just purely theoretical. So it has multiple abstract postulates, whereas intelligent design has only one. Intelligent design is not a God of the gaps argument. It is based on what we do know. For example, we do know where complex specific information comes from. It comes from intelligent designers. We do know the attributes of something that is intelligently designed and that the cell has these attributes. We do know that the cell carries these attributes. It has informational hierarchy and integrated systems, which are found from things that are intelligently designed. So it's not a God of the gaps whenever we know what things that are intelligently designed look like. We have the attributes of things that are intelligently designed, and we find these within the cell. And we also, the things that I've proposed today that would make abiogenesis scientifically implausible are things that we do know about how chemicals behave, the conditions, the temperatures and pHs that RNAs and proteins form in. These are things we know that we can actually observe and test. They break down over time. They're not compatible. Their cross reactions can occur. So this is by no means a God of the gaps, as some people say. This is from understanding that this is, that intelligent design may be the more plausible conclusion. And then prebiotic experiments for origin of life research support the need for intelligence. For example, heavy planning and intervention within the experiment. So in the papers that I have read on origin of life research, you see a lot of planning, what would go into it, what components would be needed at what temperatures and pHs to actually get this proper molecule to form. Okay, so there's a lot of planning and intervention within it. Even within the experiments, you'll literally see them like centrifuging them and removing the harmful byproducts to move on to the next step, and then controlling and regulating the pHs and temperatures to get the desired outcome. Like, so this is literally like showing the need for intelligence intervention. I mean, it would be different if they like showed, these biopolymers form from like a one pot reaction. Maybe just like changing up the temperature or whatever, something reasonable on early Earth, and then like have, biopolymers come out of this one pot reaction. But that's not what we see. We literally see heavy intervention and changing of the conditions every step of the way in these experiments. Very, very rarely is there ever like a one pot reaction that will produce something impressive, for lack of better words. And so, yeah, that is definitely needed. And then this is a quote about like how, like, if there was like two possibilities where it could either be intelligent design or life through undirected processes, he would rather choose life through undirected processes because he doesn't want to come to the conclusion of intelligent design. So here's the quote by Dr. John Wald in 1967, who is a Nobel Prize winner in medicine. Okay. So one has only to contemplate the magnitude of the task to concede that the spontaneous generation of a living organism is impossible. Yet here we are. As a result, I believe of spontaneous generation. Most modern biologists, having reviewed with satisfaction the downfall of the spontaneous generation hypothesis, yet unwilling to accept the alternative belief in special creation, are left with nothing. I think a scientist has no choice but to approach the origin of life through a hypothesis of spontaneous generation. Even though, like, okay, if you read here in the top, spontaneous generation of a living organism is impossible. They recently showed this whenever they took like a sterile whenever they like sterilized a jar and showed that like nothing living came out of it when there wasn't life already in it. Because they had this theory back then of spontaneous generation where life could basically, like, maggots could just form on meat. Like, they thought spontaneous life could just form. But no, whenever it's actually sterile, they do not see life just form spontaneously. He acknowledges this here. They found that life does not form spontaneously, but having like, even though this is the case, they're unwilling to accept alternative beliefs right here in the quote.
That quote right there is classic textbook cognitive dissonance.
Yes. So,
Beautiful
but
example.
he would rather believe that than any alternative belief. Okay, so it's like a philosophical basis that even though everything looks like the cell's intelligently designed, integrated systems, informational hierarchy, complex specific information, all of these things hint to, and push and lean us towards the idea the cell was intelligently designed. because they match the attributes of other things intelligently designed. And we cannot form a cell from bottom-up chemistry. We see chemistry moves away from life, yet we're going to continue to posit that abiogenesis is something plausible as a hypothesis. Like, this is crazy, but it seems like it's based like purely on philosophical, a pure philosophical basis.
Just like the Copernican principle in cosmology. So they have looks like they have a branch in biology where also you can't admit that humans are special and that anything that there's no God. You have to actually philosophically adopt the other position and then pretend that it's science. Yeah, that makes sense.
Yeah, and this incredulity, like it's a logical fallacy when someone dismisses a claim or argument simply because they find it difficult to understand or believe. Oh, I can't conceive of an intelligent designer. We can't test it. Okay, guess what? We also can't test. We can't test abiogenesis happening in the lab, yet you still posit it as reasonable, even though you have the benefit because you work in the naturalistic realm and this is a naturalistic explanation. You should be able to replicate it, So it just gets me fired up, to be honest. Let's see if I have anything else. No, I think this was the last slide.
So when are you going to debate someone on this is what we're going to ask you. I want to see that one live. It's going to be a break show or whatever.
I've reached out to Modern Day Debate and I've asked them like, hey, if anybody wants to like debate me on abiogenesis, let me know. But basically they can't find an opponent.
Yeah, nobody
it's hard to find a
defend this.
That makes no doubt.
Yeah.
All of this
anybody who actually knows about the state of the research, it's hard to defend. I cannot blame them.
No doubt. And all of this took me right back to the monumental debate between James Tour and Mr. Farina. And even looking back on it in memory now, that he was hitting the chalkboard with almost the same degree of specificity that you have in this presentation, minus the animations.
Yeah.
He was absolutely nailing it.
Yeah, I know. He's more of a biochemist. I do more cell biology. But man, yeah, when you actually look at the papers and the raw data, raw evidence, it is so much different than is portrayed. Literally, I was watching like on the History Discovery Channel or whatever. I don't watch like cable TV anymore, but they literally said, "Oh, you just have this chemical soup. You have some lipids, carbohydrates, and nucleotides, amino acids, and boom, life forms." Like on television, they literally said that. I was so furious. I was like, are you kidding me right now? You think you can throw these things in a jar or in any condition and watch life form? I don't care how many years you wait. I bet you it'll form away from life. They'll actually break down over time.
All beef patties, special sauce, lettuce, cheese, pickles, onions on a sesame seed bun. You
all in a puddle of tar.
For that, Tay? That's right. We got all that going on with cheese.
Yeah, going back to the tar, it's like, hey, Grandpa, this jar of tar. It's so ridiculous.
Look, we have all the components. Is it going to make life? I don't know. Hit it with lightning. Oh, I just made tar. All right, throw it out.
Yeah, dude, it's wild. It really is. And I'm sorry if it seemed like I wish it was more of a cohesive thing to like try to steelman, but it's really not. It's really all over the place. The state of the current research is all over the place. Like trying to put it together in a cohesive picture is impossible.
That slide that showed all the all the conditions where it like laid it out. It was like the one long statement. It was like, here's how we think it happened. That was the funniest thing, honestly, I've ever seen. And that's probably the best steel, man, right? Here's like, so we hit all the flags. We covered all the scenarios. We got dry cycles, wet cycles. We got this. We got low pH, high pH. We got some heat. We got some cold. We got it's in ribosome. It's in a rock. They're compartmentalized, but they're close. And we nailed it. We figured it out. It's the funniest thing.
They have different pHs in each environment. I mean, honestly, I wanted this presentation. It already took me like 40 hours at least to compile just by reading all the background research. And I tried to make it, I was like, how can I cohesively make this simple and explainable and try to like steelman the current state of the knowledge?
Delivered to you by Space Uber Eats.
Yes. And so like I wanted to add papers in here, like the most modern and groundbreaking research on abiogenesis, but it would have taken so long and been even more complex that I feel like this is already kind of complex of a presentation. But yeah, the state of the research is all over the place. They even admit this in their own conferences. But yeah, I really hope that y'all learned something and enjoyed the presentation. Sorry, I'm a little like my nose is a little stopped up and stuff, and I'm a little, stuffy, but it's okay. I hope I presented coherently and that you guys enjoyed it.
Did great. Thank you, Jen, for also jumping in there, breaking stuff down.
Heck yeah. even though he's working right now. Thank y'all so much. I guess I'm gonna end streaming. If there's any questions from the audience, let's see. Just let me know. Was there any questions that you want to ask or anything else?
I'll take a quick gandy at the chat, but while anyone's in Discord, if anyone has any questions, feel free to ask.
Said Tayler is killing it. Tayler is killing it. Tayler plausibility is all they have, and then some more Taylor's killing it statements. It looks like they just thought you did a good job.
Cool. All right. Well, yeah,
guys, there will be a
know.
test on this next week, so make sure everyone.
It's funny. Yeah, and if y'all could find anyone to debate me, I'd be more than happy to debate or take any criticism on the presentation. Just reach out to me, and I'll definitely respond.
There's a
We
strict
both want that heat.
criteria. There's a strict criteria, though. If you're going to challenge Tayler, you must have a position. The last person that tried to defend abiogenesis in a debate with you ended up saying that they didn't even have a position, and they were just kind of there to be a punching bag, I guess.
Yeah, that's so funny.
Some incoherent crap to defend, dude. I can imagine no one wants to stand up scientifically and put their name, reputation, public persona behind that garbage.
that was the debate with Grayson Hawk that you're talking about, and I watched an interview with him. He sent me an interview that he did on the scientists that did the amyloid research, and he was like, "Oh yeah, if we get the amyloid research to if we get the amyloids to then start having information that could then be transcribed to RNA, and then that could kick off the replication process." And the scientist was like, "Oh yeah, that's very wishful thinking. That's very wishful thinking, and maybe one day, but so yeah, they'll acknowledge to the people that are very zealous about abiogenesis that'll actually come defend it. They'll acknowledge the current state of the research, kind of like I showed in John Sutherland's slide. We are not even close to achieving understanding on the origin of life. That's just what it is. And for people to defend it so vehemently it's possible, it's really I would like to debate someone who actually has the understanding on the topic and who has read papers on the topic. Last night I told somebody that they were basically Christian, and they were saying, oh, maybe one day we can understand how life formed chemically, and that shouldn't ruin anybody's faith. And while I do agree with that, I told him, I said, well, if you think that we can, one day show how chemicals can form life, I think that if you're positing that as reasonable, then you maybe don't have a full understanding of the scope of the research as it is. It's because the more we learn, the further away we're getting from understanding it. It's not the other way around. And they banned me from that room just for saying that. They got super angry. So yeah. Anyway. It's an interesting topic, and hopefully I'll be coming back on here to present some of my salt flat data and surveying data I did the salt flats in the future.
Yeah, you go next Sunday?
Absolutely.
An experiment to do in Zoom because the only I'll get into that later, but I do hope to do a presentation about that in the future.
I can't wait to talk about that.
I know, me too.
Whenever you guys are ready, we will book it.
Okay.
I've been wanting to tell you thank you, Alan. From my understanding, we're the only, and it's I don't know, it's astounding. We'll have to talk about it later.
Yeah. All right, I'll stop sharing. 1 second. Okay, there we go. Yeah,
we're going to go ahead and wrap up for this episode of GLOBEBUSTERS. We'll be back next week for episode 10. We got, I think, 2 more this season after that. And then we'll be starting Monday.
Pretty bad, Ellen.
Oh, man. Shane closes out the schedule for Monday.
I don't know. What are we doing Monday? Nothing.
Model
yeah, I can't hear you. I don't know what we're doing Monday.
I got you.
Come back Monday.
Exactly. It's happening then.
We don't know what, but it's happening.
Definitely be back Monday for something. Probably some null hypothesis, Presley, maybe? We're doing that?
Check DM.
Hanging
We
out in
can
the
hear you now.
dark, just guessing?
Yeah,
Yeah,
all cleared up.
yeah, good. Well, model review, question A on Earth Awakenings, 8 p.m. Okay, that'll be awesome. Nice.
Yeah.
Put that out.
And then
Get some questions about the map or whatever.
yeah, and then that'll serve as the intro for the actual null hypothesis show. So
Nice.
then we'll be starting that the following week. But I'm sure everyone wants to hear about the model stuff and whatnot. All right, so we'll go ahead and get that going, and then we'll be back on Tuesday for probably a reading night or something. We'll see what's going on. And then the Friday community Twitter space hosted by Ken and Brian Sweet will be available to the lads and ladettes on Friday.